Next we'll have Bethany Springer presenting. She joined ACS this year, 2011. She currently attends James Madison High School, and she will be speaking about studying P2 migration after RNA passage.
They was working with a specific virus, and this is just the basic structure of the virus. This is not the virus we worked with, but this is like a really simple structure of the virus. And we were working with the core, and this is the inner core of what a virus looks like. Okay, so what is a virus? A virus is a small, non-living particle that invades and then reproduces inside of the cell. A virus attaches to a cell. It directs the cell to produce the virus's protein and genetic materials. Viruses have an outer coat that protects the virus, and then the inner core is made up of genetic materials. And the name of the virus we worked with is called a cystovirus. And a bacterial cystovirus is like this unique kind of virus because I wasn't really exposed to it before. But the inner hexagonal structure is what the other lab techs work with. So we were just trying to map out the different proteins in the core where it says RNA so that we could understand RNA packaging much better so we could better understand the virus. So the cystovirus contains proteins that carry out RNA. The P1 is the outer shell. The P2 is the RNA polymerase. The P4 is the packaging motor. And the P7 stabilizes the other proteins and makes them work more efficiently. This diagram makes it like they all point to the same thing, the P1, P2, P4, and P7. But in the models I have, like in my PowerPoint, you're gonna see the different proteins and why they're important. Introduction. Viral RNA polymerase function inside the inner capsid shell of a virus during replication and transcription. In most cells, RNA polymerases do not do that, but it's only for viruses that carry out this special function. And I like to look at RNA polymerases as how we look at DNA, so it codes for their genetic information. The real virus has a pathway where genome replication and packaging occur, and the goal of this project is to better understand RNA packaging and all the proteins involved using 3D imaging. And that's really what I was focused on, is the 3D imaging analysis of the virus. Okay. So why is this important? Like I said before, it's to provide a good understanding of RNA packaging. The virus is similar to the real virus. This can affect the gastrointestinal system and respiratory tract. It can be detected in feces. Their role in human disease or treatment is still uncertain. So this basically means that the real virus is not really linked to any known disease like cancer or diabetes or anything like that. But like in places like Haiti or like Somalia where they have certain conditions, like in suffering countries, when you leave open water or people defecate openly, these diseases tend to transmit, and that's where the real virus comes in. So methods. Using cryo-electron microscopy, the core of the virus was suspended in viscous ice. This transmission electron microscope was used to obtain high magnification and high resolution of cells. We use the Tech 920, but even with such advanced microscopes, when you get the scan, it's still very hard to make out each protein and get it as specific as I wanted it. a collection of images were scanned to the computer for the analysis of the Cisco virus. So the 3D imaging process, we use this program called Linux to better map out the proteins, and we use programs like iMod, ImageViewer, Chimera, and all those 3D EMs. So the first thing I did was determine the transfer function for the microscope, and then we did a pre-processing step, which takes the microscopy conditions and writing them into DAT files. All this really means is that after we get the scans of each like core of the virus, all we're trying to do is like remove any blemishes or unwanted gray matter so that we can box them out correctly, and that's the next step. All of the particles have to be boxed out manually, so you have to click there, sit there and click every particle as it comes, and you also have to be mindful of certain conditions. So this is what a basic command looks like. I didn't have to enter this entire thing into the computer. I just entered the second line. So where it says auto PP, I entered that in, and then it says enter your choice 3. So then all these things come up, and I wasn't used to Linux at all, so this took me extra long to understand what it was even talking about. But I had someone to help me, obviously, but it was still a bit challenging. So this just really says, like determining the voltage, like all these conditions are the microscope conditions. So you have to be aware of the microscope conditions because it later on affects your results. And this is boxing. Boxing is a difficult task only because you have to be aware of certain things. For example, the images, the particles that I were able to box out had to be like separate. They can't be too close together. You have to be mindful of any like unwanted gray matter. That's why like in the upper, well, in my upper right hand corner, there's like a lot of, so like over here, there's like a lot of like clump particles. You can't use any of that. So even if on a regular scan we get like about 2,000 particles, by the time I'm done boxing everything, I might only end up with like 200. And I need at least 1,000 particles to make this experiment even like worthwhile. So I had to like sit here and box each one correctly. So I ended up boxing like 1,000 for this one. And so it was pretty good because then I was able to get some decent results. And this is a montage. The montage basically just shows me in order what I boxed, and some of them are kind of slanted, so the next couple of steps helps me to like orient them around the same origin so that when I need to construct my model, they'll all be identical. Because if you remember a couple of slides prior, there was the hexagonal core, so I need all of the all the particles to be almost identical to the core so that it would be as close to as possible to the little thing. And then the next step was just some more additional imaging processing. We are normalizing all the box particles so that they have the same standard deviation and average. And then this leads to the initial reference model. So this is what I ended up with. This virus is called PLM687 unexpanded, and this is under high resolution. And this, even after boxing over 1,000 particles, this is like a okay image, because other papers show that you need a resolution of under 10, and my resolution was not under 10. But from here we can see the outer core, which is P1, the shell right here. Okay, it's not moving. Okay, right here. This is the P1, this like dark gray lining, and then the P4 is right here, and the P4 is going to be the packaging motor, which basically just like rushes in and does what it has to do. And then inside right here, I thought this was P2, but then you have to do additional steps to prove what each protein is. So one of the questions I had to ask myself is basically, what is this central density? So my results. The highlighted numbers are the only important numbers. The 18.75 is my resolution, and that's too high. So even as good as that image is, it's still, I need a resolution under 10. And the 1023 just shows the number of particles. So there were a couple of tries where we do this like every week. We get the scans and we perform this every week. And one time I only had like 200 particles, so I had to like throw away that because it makes no sense to like make an image if you only have 200. You need at least 1,000 or more. So discussion. What do we know? We know the P1 is the outer shell, the P2 is the polymerase, and the P4 is the packaging motor. So I just want to know where was this P7, because the P7 is supposed to stabilize all the other proteins. So I figured it would be somewhere obvious because it plays such an important role, but I still don't know where that is. Another question was how should we interpret the central densities? In some of the scans we received, we thought that P2 was the central density, but then in others there is no central density, so we just didn't understand what that was supposed to be. And then the last question was how does P2 change position once the particle packages RNA? So my future work involves this program called Euler, which is it's actually spelled Euler, but it's called Euler. And what that's the program, but the software I use is the parallel orientation refinement. And his belief was that you could any image expressed on 3D space can be like defined by at most 3 angles. So when I'm trying to be extremely precise and finding certain proteins, I use this so that I can know exactly where it is and then compare the images, and then I'll be able to further understand what the proteins are. This line is the microscopy conditions, and it just shows things like voltage and other things that I have to be aware of, so I know if it's too high, then I have to change certain things from the biological standpoint. And the blue angles down here are the angles that show you the precise location of the proteins. When the images are combined, it creates 2 models showing the location of different proteins. So when I input this into the computer, I'll